So the title of my talk is Long-Term Sustained Growth of Insulin-Producing Cells using a scaffold that we made, which is made up of agarose and silver-coated chitosan. Okay. The silver is in nanoparticles form. And then we tried to grow mice pancreatic cells on them for about a month or so, like 30 to 40 days. And we saw that it was producing insulin. And we wish to take this forward as a tissue engineering application in future as a pancreatic substitute or a transplant. Okay, so this is where I come from. This is where I work in Goa. And this is BITS Pilani Goa campus. So I'll just tell you the take-home messages of the abstract that I submitted. So I'll not talk about diabetes too much because everybody has been talking about it. So I'll just hold on a minute. There's a lot of echo. So yes, thank you. So diabetes is a group of diseases characterized by high levels of blood sugar for extended periods of time. We all know that. And there are a lot of therapy, a lot of medications available, but there are also challenges. So my work is on regenerative medicine, and now it's coming up in a big way. So I'll just tell you what I have worked on. So we synthesized an agarose-chitosan coated silver nanocomposite scaffold, and we isolated pancreatic cells from bulb C mice, and then we grew them together on the scaffold, and we waited for about a month, and we tested its effectiveness in terms of insulin secretion. The methods we used were to stain, if we have to isolate, islet cells, we stain them with dithizone, and we do gene expression studies, real-time PCR, Western blotting, protein profile, and also for some markers and some microscopy with flow cytometry and microscopes. So the isolated population that we took out from the bulb C mice were found to be positive for glucagon, PDX1, PAX1, and there was a 200-fold change in the transcript level of insulin. The cells, upon seeding on the scaffolds, exhibited sustained growth and insulin secretion as confirmed by Western blotting. Overall, the study demonstrated the suitability and application of agarose-chitosan silver nanoparticle composite for pancreatic tissue engineering. Now I'll show you my results. So these are the results of dithizone staining. So characterization of mice primary pancreatic cells using the dithizone staining. This was performed to isolate pancreatic cells which showed red stained beta cells. Now this is the gene expression profile, characterization of mice primary pancreatic cells. So these were the relative expression of various pancreatic genes after 4 days of primary culture. So we checked them for glucagon, amylase, insulin, and PDX1. So gene expression was relative to GAPDH. RT-PCR shows positive but varying expression of the markers. The experiment was repeated, and the normal representation of the average was given. So this is the Western blotting profile of the same markers: glucagon, PDX, PAX4, and insulin. And this is in flow cytometry. So characterization of mice primary pancreatic cells after 4 days. And as you can see, there is a slight change in the expression pattern of PDX1 and insulin. So it's coming up. So 22% for PDX1 and insulin is 27.8% compared to the unstained population of cells. So now we'll characterize the artificial construct with mice pancreatic primary cells. So cell viability is very important. If you are growing cells in a different environment with foreign, constructs and composites, so you have to be first sure that the cells are biocompatible, the cells are alive, they're proliferating and happy. Otherwise, you cannot grow them for a long time. So first thing we did was the MTT assay. So this is a test in which, if you have more cells, at the end of it, it turns formazan crystals into purple solution when you add DNSO. So it's a good, very common classical test, so which we did for cell viability and proliferation. So if you see here in the plot, mice pancreatic primary cells were grown on the scaffold, and they showed prolonged sustained growth even on day 40 as compared to 2D controls. So the 2D controls are in green, and the ones that are grown as 3D like cultures with the scaffold are in red. So after a few weeks, the composite cultures do better than 2D cultures. Now, at all points of time, you have to make sure that the cells are there. So what you do is you stain them with DAPI so that at least you can see the clear nuclei of the cells. So just to make sure the cells are still there. Many a times, if the experiment is not done correctly, the cells may not grow on the scaffold and they will grow on the substratum of the Petri plate. So this is the microscopic analysis of mice pancreatic cells growing on the scaffold. Immunofluorescence staining showed the locations of the different markers like glucagon, PDX, and insulin. So this showed that there is presence of insulin even on day 30. These are the qualitative analysis of insulin secretion with time. Western blot was performed on spent media to check for the expression of insulin on days from day 5 to 40. So we did like only media was the negative control, and then we checked for insulin on spent media day 5, 10, 20, 25, 30, 35, and 40 days. So similarly, we did other tests also, and the expression was seen under different glucose conditions as well. So the summary or the take home is that we successfully made a pancreatic tissue construct which could secrete insulin over a period of 40 days. Now we need to go to animal studies, clinical trials, etc., to see how it does in the future. So this is the summary slide in a nutshell, what we have done so far. So here you have the scaffolds. They're like spongy with holes in them. And we use different techniques like freeze drying to make them so that they can access, media can go inside for cells to populate these structures. We took mice, we took out the pancreas, we did collagenase treatment, made primary cells. As I showed you, we characterized the primary cells with these protocol for Western blotting, quantitative PCR, dithiazone staining, and flow cytometry. And after that, we grew them with the composites for roughly around 40 days, and we characterized it again through similar methods. like MTT assay, immunofluorescence, Dumpy staining, and Western blot. So I think that is, I will not go into any more details because otherwise it gets really boring with all the jargons and all. I'm happy to be here, and I liked the talks earlier on. And if you have any questions, you can definitely email me or just go around and read the paper that we published. So it'll be much more clear. And we are very grateful for our funding from DST, Department of Science and Technology, India, and the Indian Council of Medical Research for funding. And these are the people who worked with it. Nupur Kumar is my PhD student who did the work. Now she's doing a postdoc in Sweden. And we had collaboration with Professor Sonawane's lab in Indore, and Padhi Abhinash is his student. So they also helped us with the animal work. We did the scaffold at Goa, and then we sent the student and the scaffold to Indore to do the rest of the work. So I thank you all for listening to my work, and I'll be happy to answer any questions that you might have.